plenty of time for questions. Would that work?
Brilliant. That sounds excellent. That sounds great.
And
thanks.
Will most of the people at least have sort of a undergraduate level biology background?
Yes, absolutely.
Okay,
Yeah.
People,
yeah.
So most of the people know what DNA and RNA and proteins are?
Yes, definitely. Okay. Yeah. Great. Brilliant. Thank you.
Thank you.
Paddy, are we all right to get started?
Yeah, I'll just keep admitting people as we go.
Okay, great. Should I hold on a minute, do you think?
I'd say maybe just give it 1 more minute because they're flying in at the moment and it's literally just gone 1 o'clock.
Great, sounds good. Okay, well, we're good to go?
Yeah, I'd say so.
Yeah, I'd say get off there now.
Brilliant. Okay, great. So good afternoon and welcome back to all to everyone here again today and to all of our attendees joining us from the States. Good morning. I'm thrilled and to be honest, my name is Maeve Heaney again, and I am chairperson and co-founder of the Translational Medicine Society. So I am thrilled and to be honest, it's still slightly surreal to present today our next speaker and keynote speaker, Dr. Dan Barouch, for the inaugural Translation Medicine Conference 2021. So Dr. Barouch is the director of the Center for Virology and Vaccine Research at Beth Israel Deaconess Medical Center, a professor of medicine at Harvard Medical School, and the lead virologist and researcher in the Beth Israel Johnson & Johnson vaccine candidate, which is based on an adenovirus design pioneered by Dr. Barouch and his colleagues some 10 years ago. So Dr. Barouch has transformed healthcare with his work focusing on tackling and conquering infectious diseases such as HIV, TB, Zika virus, and now the coronavirus. These viruses clearly have detrimental effects worldwide, and tackling them by vaccine is arguably the most definitive approach, which is clear to us all now given the current climate. So Dr. Barouch, I believe it is fair to say you show unmatched drive to fill the gaps and find solutions to combat these viruses that threaten global public health. From opening up your first lab in Harvard in 2004 and beginning with the goal to find a vaccine against HIV, to now tackling perhaps the largest challenge to face our generation, SARS-CoV-2 virus. Elected in October to the National Academy of Medicine in the US for his well-deserved recognition of his leadership and major contributions to advancing human health and medical science, I am delighted to have Dr. Barouch here to give us insight into his journey and developing a vaccine during a pandemic. So I'll pass it over to you. Thank you very much, Dr. Barouch, for joining us here today.
Well, thank you very much for those kind words. Let me try to share my screen first. Do you see my slide and can you hear me okay?
Yeah, everything's perfect. Thank you.
Okay, great. Well, first, thank all of you for organizing. I understand this is really a student organized and student run conference. So congratulations to you folks for your interest in this area of translational medicine. And I hope that at least for many of you, your interest in being here today translates into a lifelong interest in helping to tackle some of the pressing global health problems of our time. What I'd like to do, I understand that we have just under an hour together today. What I'd like to do is give you some background into where we are in COVID-19 vaccine development as a whole, but also concentrate from a storytelling perspective, really what we've done over the last year and how we hope this might be able to contribute to what we're going to see during 2021, which is the most massive global vaccine campaign in history. So This project started for us on January 10th of the year 2020. So exactly a year ago, a few weeks more than a year ago today. I remember that date because that was actually the date of the annual Barouch Lab retreat, which we held in the Museum of Science in Boston. And I get together with my lab group once a year. We all huddle together and talk about the work that we've done over the last year and make plans for the future. And during that meeting on January 10th of last year, we talked about the reports over the last 2 weeks coming from Wuhan in China about a new mysterious pneumonia that was appearing. And on January 10th, there were reports that there were 41 cases and 1 death. And we found those numbers to be alarming. Just think about it, a year ago, 41 cases and 1 death was alarming enough that half a world away, we were discussing it, and we felt that a vaccine was potentially going to be needed. Similar discussions were happening probably in dozens of groups around the world on that same day. Also on January 10th, the SARS-CoV-2 sequence was released that evening by the Chinese researchers that basically was the starting point of the global vaccine effort. So fast forward a year, we've gone from 41 cases to over 100 million cases. This slide is about 2 days old, and I believe yesterday was the very sobering milestone of reaching 100 million global cases. So we've gone from 41 cases to 100 million cases, and we've gone from 1 death to over 2 million deaths in 1 year. It's truly astonishing how a virus can rip through society, basically go to every corner of the world, and basically interfere with all of our professional and personal lives in such a profound way. The scientific response has been astonishing in many areas in understanding the basic virology and immunology of infection, the development of therapeutics, science-driven public health measures. And for vaccines, which is arguably the most important medical interventions in history for viral pandemics, the WHO has estimated that there are over 200 vaccine candidates under development. It's truly astonishing if you think about it, that with a new viral pandemic, over 200 groups are developing vaccines. These really span all of the platforms you might expect: DNA, RNA, protein, vectors, inactivated viruses, even cell-based vaccines and other vaccine modalities. I would argue that the timeline to show clinical efficacy in clinical trials is actually not the most relevant timeline. The most relevant timeline is the timeline to produce and deploy billions of doses of vaccine worldwide. In the United States, the first vaccines that have been approved under emergency use authorization are the mRNA vaccines from the companies Pfizer and Moderna. There's also there's an inactivated virus vaccine approved in China. There's the chimp ad ox vaccine approved in the UK and other countries, and several others worldwide. These vaccines are currently being rolled out under emergency use authorization, and we hope that additional vaccines will become available over the next few months. So the COVID vaccine field took a giant step forward on November 9th, just about 2.5 months ago, when Pfizer and BioNTech announced the results of the world's first phase 3 vaccine trials. And this new vaccine technology called mRNA vaccines was shown to be highly effective at preventing symptomatic COVID disease. Just a week later, Moderna announced very similar results with a very similar vaccine candidate based on RNA. And just a week after that, AstraZeneca with the University of Oxford reported that their chimp ad vaccine also showed substantial efficacy in preventing COVID disease. So November was quite a stunning month. And then in the United States, the first 2 RNA vaccines were approved by the FDA for emergency US authorization and rollout. So these are the 5 vaccine programs that are either in or finished phase 3 clinical trials in the United States. There's about 10 to 15 globally, but these are the 5 that we're developing here in our country. The Moderna and Pfizer RNA vaccines. These phase 3 studies were launched in July and showed efficacy in November and were approved in December. The AstraZeneca chimp ad ox vaccine has completed or has read out phase 3 trials in the UK and Brazil. The US-based study is still ongoing. It was launched in August and will hopefully read out in the next month or 2. The J&J vaccine, which is the vaccine that we've been very closely associated with, is based on a human Ad26 vector. Phase 3 studies were launched in September and are scheduled to read out in January. And if you realize, it's January 28th. And so we're expecting the results of this trial literally any day now, not as of today. And finally, the 5th vaccine study that the United States is pursuing is a Novavax vaccine, which is a purified protein vaccine. And that trial launched in December, and efficacy is hopefully going to be in the springtime. I should say globally, these similar technologies are being pursued globally, with the ad vectors being pursued in China and Russia, as well as the AstraZeneca vaccine tested in the UK and Brazil. And in addition, in China, there's 3 or 4 inactivated virus vaccines that are also being tested in phase 3 clinical trials. So the mRNA and the ad vector-based vaccines are gene-based vaccines, and these have emerged in the US and to a large extent globally as the lead vaccine candidates because of their speed, their flexibility, and their efficacy. So mRNA vaccines, together with some colleagues at Moderna, we have a review article that's going to come out in the journal Cell in March. And so these are some figures that we've just finalized for this review article. So mRNA vaccines, essentially, the way they work is you have a vaccine antigen, like the coronavirus spike protein. You take the amino acid sequence, and from that, you derive the RNA sequence. The RNA is then manufactured, and it's formulated in lipid nanoparticles, essentially fat droplets. And that's essentially then put into the vaccine vial and injected into the arm. The fat droplets essentially help protect the RNA and help get the RNA into cells. Adenovirus vectors conceptually is very similar, except that it's DNA instead of RNA, and instead of a fat droplet helping to get the nucleic acid into cells, it's an adenovirus particle. So the way adenovirus particles are created is they're vectorized. So the replication machinery is taken out. So they're replication defective. They can't replicate in humans. They don't cause disease. And an antigen of interest, such as the coronavirus spike protein DNA, is then stitched into the adenovirus particle. The DNA of the adenovirus particle here in step 3 is used to transfect cells, and viral seed stocks are then grown up, and then viral particles are purified. The purpose of the viral particle is to help get the spike protein DNA, here shown in blue, into cells. So RNA vaccines and adenovirus vectors are conceptually very similar. Basically, there is a delivery system that help gets the nucleic acid of the gene of interest into cells. And then the host makes the protein, and the host makes an immune response to the protein. So vaccine development for a new pathogen usually takes many years, if not decades. So how is it even possible to develop a COVID-19 vaccine, or I should say multiple vaccines, in just 1 year? That's truly unprecedented in the history of vaccinology. And I think what we're seeing over the last year with not one, but multiple vaccines, either approved or possibly soon to be approved in approximately a year, is really a testament to advances in immunology and vaccinology that have occurred over the past 2 or 3 decades. So we're really seeing the fruits of many, many years of scientific advances in multiple fields, essentially converging on trying to solve a global problem. Essentially, the strategy is to use vaccine platforms that have been developed for speed and precision, particularly these gene-based vaccines, and platforms that have already been tested for other pathogens. So basically platforms that are new and quick and can be developed fast, but not so new that they've never been tried before. And hence the field has sort of settled onto RNA vaccines and advectors, at least for many of the current global vaccine programs. In addition, manufacturers perform development activities in parallel rather than in series. So for example, all the major companies that I mentioned started to mass produce their vaccines and millions of doses while clinical trials are still underway, with the idea that if approval is granted, then there's millions of doses to distribute, essentially starting immediately. Now, never before in history would that be considered acceptable, because that's just too much of a financial risk. Because if the trials fail, then literally millions of doses of vaccine are simply thrown in the trash. So there has to be substantial tolerance of programmatic and financial risk. But financial risk is okay. But what cannot be risked is patient safety or regulatory integrity, because a vaccine ultimately needs to be trusted by the public, otherwise it will fail. Also, for vaccine development, mass production and global distribution is required, because the goal is to achieve herd immunity to end the pandemic. So we started in January of last year, COVID vaccine effort, really on 2 parallel trajectories. One was a basic research trajectory to try to answer basic research questions that would apply not just to our vaccine candidate, but to all vaccine candidates. And secondly, to help develop one specific vaccine candidate, which we hope will eventually be useful. So in terms of basic research, we started to ask questions, basic science questions that we thought would be very relevant for vaccine development right at the start of the pandemic. And the 2 questions that came to mind is there evidence of natural protective immunity? And in particular, will individuals who recover from COVID be protected against re-exposure? And second, is there vaccine-induced immunity? And if so, what are the immune correlates of protection? These are human questions, and answers are filtering in from large-scale human studies. But we wanted to get an early definitive answer, at least from a conceptual point of view, to these questions. And so we developed a rhesus macaque model of SARS-CoV-2 to answer these questions. And our initial answers to these questions, at least in animal models, were published in May of last year. In the first study, we developed a SARS-CoV-2 model in monkeys. And we infected 9 monkeys with 3 different doses, a high dose in group 1, a middle dose in group 2, and a low dose in group 3 of SARS-CoV-2 by the intranasal and intratracheal routes. And here we show virus in the lungs following infection. You can see in all of the animals, they were infected, and they had virus replication in the lungs for about a week or 2. They also had virus replication in the upper respiratory tract, peaking on around day 2, and then resolving by about 3 to 4 weeks. And if we look at pathology in the monkeys following infection, then basically there's a very clear pathology of viral pneumonia. So on 2 or 4 days following challenge, we see in panels A and B that there is dense inflammatory infiltrate. In panels C and D, you can see sloughing of epithelial cells and terminal bronchi. In panel E, you see a multinucleated giant cell. In panel F, you see hyaline membranes in the lung. In panels G and H, you can see immunohistochemistry staining for SARS nucleocapsid, showing infected multinucleated cells and an infected pneumocyte in H. I is RNA scope analysis, showing the presence of SARS-CoV-2 RNA in cells in the setting of dense inflammation. And J, K, and L shows a large bronchus, showing inflammation in J and dense infected cells in K and L by RNA scope and by immunohistochemistry. But the experiment that we did was a re-challenge experiment. The 9 monkeys I showed you earlier that were infected, these animals showed resolution of virus replication by about 3 weeks. And then we waited until day 35, and then we re-challenged them with a second inoculation of virus, and there was very robust protection. So this was one of the first examples of natural protective immunity, which of course we thought would be very relevant for all vaccine programs. In the second study that we published last May, it was our first vaccine study using prototype DNA vaccines. Now, these were not the vaccines that we ultimately took into the clinic, but rather were early prototype vaccine constructs where we wanted to get a rapid assessment of what vaccine protection might look like. And so we vaccinated monkeys with these DNA vaccines expressing different forms of the spike protein, and we challenged the monkeys with the same model that I just told you about. And this is peak viral loads in lung following challenge. You can see the sham controls on the left all had a high amount of virus, and the different vaccines showed various levels of reduction of that virus. But even these early prototype vaccines showed a reasonable level of protection. And that made us optimistic about the potential of more potent vaccines coming up. We also noticed that there was an important correlation, that the higher the vaccine-induced neutralizing antibody titer was, the lower the amount of virus in BAL and nasal swabs. So essentially, the vaccine-induced antibodies correlated with protection. And we reasoned that such a correlate of immunity, if it were generalizable, and if it were also applicable to humans, would be a very powerful tool that would accelerate vaccine development. So the vaccine that we did take into humans was based on a human Ad26 vector. Basically, this is a replication incompetent, a deactivated common cold virus. And we initially vectorized Ad26 in 2004 for our HIV vaccine program. So we basically made this vaccine platform not for SARS-CoV-2, but for HIV. And we've developed this vector for HIV vaccines over the last 15 or 16 years. And in fact, our phase 3 trials for HIV are still ongoing. We then used Ad26 in 2016 for our first pandemic response to Zika virus. And you could say that was almost maybe a dry run for what happened last year. And for Zika virus, we showed that even a single shot immunization with an Ad26 Zika vaccine raised neutralizing responses in essentially almost all individuals. And our colleagues at our industry partner at J&J developed an Ad26 Ebola and RSV vaccines also, which led us to learn a lot more information about this vaccine platform. Overall, pre-pandemic, Ad26 was already shown to be safe and immunogenic in over 110,000 people to date. And last year, J&J received regulatory approval for their Ad26 Ebola vaccine in Europe. So there's regulatory experience with this vaccine platform. And with the Ad26 RNA vaccine, there's also pediatric experience into babies down to 6 months of age. So we think that if this vaccine does prove to be safe and effective in humans, then age de-escalation into children should be very possible. And it would be great if we can vaccinate the entire school-age population of children before school starts in the fall. Also, from the Ebola experience, J&J was able to ramp up production so that they have readily available methods for manufacturing millions of doses. And also, they developed a formulation that does not require freezing, because they needed to deliver their Ebola virus vaccine to Central Africa. So we know that this platform has the potential for a single shot vaccine, mass production, and no sub-zero freezing. And we thought these properties might be useful for a pandemic response. So these are data not from the Ad26 COVID vaccine, but our Ad26 Zika vaccine. And I'm showing you these data because it shows you how different fields really build on each other. So first, without our HIV research, there would be no Ad26 based COVID vaccine that is about to read out in clinical trials. From the Ad26 Zika virus vaccine, this is a clinical trial where we tested, and these data will come out in a few weeks. We tested a low dose and a high dose of the vaccine, either as a single shot vaccine or as a 2-shot vaccine. And as you can see here, virtually all recipients developed neutralizing antibody responses within 2 and higher responses after 4 weeks after immunization. And even a single shot vaccine had neutralizing titers that lasted for more than a year. A second shot in 2 of the 4 groups increased these titers by more than tenfold. So we think that the Ad26 Zika vaccine is immunogenic as a single shot vaccine, has durable antibody responses that last a year, and these responses can be boosted by a second shot. So these basically formed our thought processes when we started to think about how to make a COVID vaccine. So this is actually what we did. On January 10th of last year, as I mentioned, the virus sequence was released and we started analyzing the sequences. And so we worked over the weekend so that on Monday, January 13th, we were ready to start building synthetic genes to design the actual vaccine constructs. By the end of January, we had a collaboration agreement with our industry partner, J&J, with whom we work closely for HIV and Zika. And obviously, J&J had the manufacturing expertise to make these vectors at high quantities. In February, we immunized mice and rhesus monkeys with early prototype vaccine constructs. By the end of February, we developed a virus challenge stock. In March, we started to establish small and large animal models of disease. And by the end of May, we published our observations on natural and vaccine immunity. By the end of July, we started our phase 1/2 clinical trials, and we also published the ability of a single shot Ad26 vaccine to protect non-human primates. And just less than 2 months later, we started our first phase 3 clinical trial, which was a single shot vaccine in 45,000 individuals. And this is the vaccine that's going to read out any day now. And in mid-November, we started a second phase 3 study of a 2-shot version of this vaccine in 30,000 individuals. And that trial is still enrolling. So this program is sort of on the brink of finding out whether or not this vaccine is effective in humans. If it is effective, and I can honestly say as of today, I have no idea because the studies are still blinded, but we will all know in a few days. If this vaccine is found to be effective in humans, then J&J is in a position to rapidly apply to the US FDA for emergency use authorization, and I presume to the European authorities very shortly after that. And if granted, if approvals are granted, then J&J does have a commitment to producing at least 1 billion doses of vaccine for a global rollout during this calendar year. So I'll show you a few slides from the paper that we published in July showing that a single shot Ad26 vaccine can protect rhesus macaques against SARS-CoV-2. So what we did together with our J&J colleagues, we produced not 1, but 7 versions of this vaccine expressing different spike protein immunogens, including versions that include different leader sequences, full length versus deletion mutants, as well as native sequences, as well as sequences that were modified with a furin cleavage mutation and a diproline stabilizer motif described by Jason McClellan and colleagues. I'll just refer you to this last one, S.pp, that refers to a native leader sequence, a full-length spike, and the 2 proline stabilizing mutations, because this is the one that ultimately was most effective in monkeys and was taken into humans. So the study we did involved 52 rhesus macaques, mixed gender. We did a single shot vaccine because of our experiences with Zika, in which we gave 10 to the 11 viral particles of Ad26 vectors expressing 1 of 7 of the spike protein variants, and there was a large control group. And then 6 weeks later, we did a challenge with 10 to the 5 TCID50 of SARS-CoV-2 by the intranasal and intratracheal routes. These are neutralizing antibody titers induced in the monkeys. And by week 4, all monkeys developed antibody responses. And the SPP vaccine shown here generated the highest responses. And based on these data, then this was prioritized for clinical development. Following challenge, here we show viral loads in the lung, in bronchoalveolar lavage. In the sham control group, shown on the left, all 20 animals were infected and had high levels of virus in the lung, peaking at around day 2 and generally being resolved by about 10 days, similar to humans. In the SPP vaccinated groups, there was no virus detectable at any point in time, so there was complete protection in the lung. The other 6 vaccines that we tested also showed reasonable protection in the lung with 0 to 2 breakthroughs per group. And the breakthroughs that were seen were still lower in magnitude and shorter in duration than in the control group. In nasal swabs, it's typically harder to protect against virus replication in the upper respiratory tract. In the sham controls, we saw high levels and even in some cases prolonged virus replication in the upper respiratory tract. In the SPP vaccinated animals, 5 out of 6 were completely protected and 1 showed a low blip of virus. So there was protection in the upper respiratory tract as well. There was also partial protection with the other 6 groups with 1 to 3 breakthroughs per group. But as you can see, protection is lost more easily in the upper respiratory tract compared to the lower respiratory tract. These are, this is a summary of protective efficacy. These are peak viral loads in the bronchoalveolar lavage on the left and nasal swabs on the right. The sham controls have high levels of virus of 5 or 5.5 logs in essentially all of the controls. And there was differing levels of protection seen with the vaccines. The SPP vaccine shown here gave complete protection in the lung and near complete protection in nasal swabs. We were also able to confirm the correlates that we saw with the DNA vaccine, in that at week 2 and even better at week 4, the neutralizing antibody titers raised by the vaccines inversely correlated with peak viral loads here in the lung, and not shown here also in nasal swabs. Together with my colleague Galit Alter at the Reagan Institute, we also looked not just at neutralizing antibodies, but a whole host of other antibody parameters, binding antibodies, different subclasses, different functions. And what we found is that many of these antibody correlates actually tracked with protection. We divided the animals into 2 groups, the animals that were completely protected and the animals that were only partially protected. And the best discriminating factor between these 2 groups was the neutralizing antibody titer shown here. But close behind were other antibody functions, such as antibody-dependent NK cell activation and antibody-dependent cellular phagocytosis. And so we can't really distinguish which antibody function is most important in the study because many of these antibody parameters correlated with each other as well. But our going hypothesis is that the neutralizing titers are probably most important, but that other antibody functions might also contribute. So this study showed that a single shot Ad26 vaccine resulted in complete protection in the lung and near complete protection in the upper respiratory tract. We reasoned that a single shot vaccine in humans could have major advantages over a 2-shot vaccine for mass immunization and pandemic control. But we also reasoned, and now we have shown, that a second shot of this vaccine can increase neutralizing antibody titers substantially. So we thought that clinically it's very important to test both a single shot vaccine and a 2-shot vaccine. And that's what's happening. So neutralizing titers were the strongest correlate of protection consistent with a prior DNA vaccine study. And so the development plans for this vaccine, which has been renamed Ad26-CoV-2S, so that's the name you might see if you read about this vaccine somewhere. Phase 1/2 studies were initiated on July 22nd in 1,000 individuals. And the first phase 3 efficacy study was started just 2 months later on September 21st. So before telling you about the clinical program, I'm just going to review one more basic research study. I guess this would be in the category of basic research studies that really apply to all vaccine programs. And we thought that it was very important to have a deeper understanding of correlates of protection. Most importantly, correlates of protection are important for our understanding of viral pathogenesis and immunity. But a better understanding of correlates actually has some very important practical uses as well, because correlates of protection might be needed for future approvals of vaccines. Because as vaccines are rolled out to the general population over the course of the next couple months, then it will become practically impossible to conduct placebo-controlled efficacy trials, both ethically impossible and practically impossible. And that's happening now. So basically, moving forward, it will simply not be possible to do placebo-controlled efficacy trials of vaccines, certainly not in the US and in Europe. So given that we all think that there should be additional vaccines developed, either the current vaccines against some of the viral variants or new vaccines that might show additional beneficial properties, then approvals based on immune correlates is likely going to be the way forward. Also, immune correlates might be useful to help us understand waning protective immunity following either vaccination or infection. So we published this study about a month and a half ago in which we tried to define the correlates of protection in the more tractable model of SARS-CoV-2 infection in rhesus macaques. So our previous vaccine studies that I just mentioned show that there's a statistical correlate between antibody responses and protection. Those vaccine studies do not prove that antibodies are important for protection. It's just that they statistically correlate with protection. In the context of vaccine studies, in humans or in animals, you can't really define the mechanism of protection because the antibodies that we measure are really in the context of innate immune responses, T cell responses, and many other immune perturbations. So to actually prove whether antibodies can protect, we actually did a classic adoptive transfer study. We purified IgG from convalescent macaques, and we determined whether this purified IgG could protect naive animals. And if so, then that would be a formal demonstration that antibodies protect. And if we can do that, then we should be able to define the neutralizing titers required for protection, at least for this model. So 3 days prior to challenge, we transferred purified IgG into 12 rhesus macaques at 3 different doses, 250, 25, and 2.5 mg per kg as a single infusion. And then 3 days later, on day 0, we challenged the animals with 10 to the 5 TCID50 of SARS-CoV-2, just like before. Here are the neutralizing titers in these animals. So on day -3, the day we started, I started with undetectable neutralizing titers. By day 0, which is 3 days after the infusion, we detected high titers of about 500 in the animals that received a high dose, and these titers then dwindled over the 14 days post-challenge. In the mid-dose group, at 25 mg per kg, there was almost a mathematically exactly 10-fold decrease in the neutralizing titer of about 50. And these also declined over the 14 days post-challenge, although you could imagine maybe one of these is ticking up. In the lowest dose group, they had undetectable neutralizing titers prior to challenge, and came up strongly by day 14 following challenge, very similar to the control group. So these are the protection data. So the high dose group fully protected. The mid dose group, there was 1 breakthrough. In the low dose group, all 3 animals were infected, but appeared to show a shorter duration of virus compared to the controls. In nasal swabs, we saw a very similar outcome. The high dose group fully protected. In the mid dose group, there was 1 breakthrough. In the low dose group, all 3 animals were infected, but generally had lower magnitudes and shorter duration of virus replication compared with the sham controls. So with a logistic regression analysis, we tried to define the neutralizing antibody titer threshold for protection. And in this model, it was a titer of about 50, which actually was quite low. And we wrote in the paper that this titer is relatively low and should be achievable by vaccination. And therefore, we felt this was very promising for vaccines. And we actually wrote that paper before the November 9th announcement by Pfizer and BioNTech. The second study that we did in this paper was a CD8 depletion study to really ask the question of what is the role of CD8 T cell responses in protection against SARS-CoV-2 challenge. Now, I just showed you that antibodies alone can protect. So what's the role of CD8 T cells? So we decided to use a different model. So we used the model of SARS-CoV-2 re-challenge in convalescent macaques. So what we did is we looked at infected animals, we held them for a while until their natural antibody titers from convalescent infection dwindled, and then we did a CD8 depletion. We can do that in monkeys by an infusion of a monoclonal antibody against CD8. And so 5 animals received this depleting CD8 antibody, which essentially results in complete total body CD8 depletion. and the other group received a sham control. And then in these depleted animals, we did a re-challenge with the same 10 to the 5 TC8050 of SARS-CoV-2. So these are the antibody titers from the study. The re-challenge animals that were used in the adoptive transfer study are shown here. 4 weeks after a primary infection, titers are generally about 100 or 150 or 200. If we wait another 3 weeks, these titers start to dwindle so that the majority of them are under 100. And so these are animals that we thought might start to become susceptible to re-challenge. So here are viral loads following the re-challenge and bronchoalveolar lavage. The CD8 depleted animals still had very good control of virus with only one blip, and the sham controls were protected. But in nasal swabs, we observed that the CD8 depleted animals showed breakthroughs in 100% of animals, compared to only 1 out of 5 in the sham controls. And you can see this is a significant difference with the CD8 depleted animals showing breakthrough much more than the sham controls. And this basically demonstrates that CD8 T cell responses really do play a role in protection if neutralizing antibody responses are waning or subprotective. So this study shows that purified IgG, so purified antibodies, are sufficient to protect macaques against SARS-CoV-2 challenge in a dose-dependent fashion. We also showed that the threshold neutralizing titer for protection, at least in this model, are low at around 50. And this appears to be very similar to what we've seen in the vaccine studies in humans. And we thought that these relatively modest neutralizing titers should be achievable by vaccination. And indeed, the subsequent data showed that they were. We also showed that CD8 depletion reduced protection against re-challenge in convalescent macaques with waning antibody titers. So this essentially immunology study showed that neutralizing antibody titers alone can protect, but cellular immune responses also contribute to protection when neutralizing titers are borderline or subprotective. So I'll turn now for a few minutes to the clinical program. The interim results of the phase 1/2 trial of the Ad26-CoV-2S vaccine were published. Well, the early look data was posted on bioRxiv in September, and a more complete data set was published here in the New England Journal of Medicine several weeks ago. And basically what it shows is that a single shot vaccine raises binding and neutralizing antibody responses, as well as T cell responses in virtually all vaccinees. The way the study was arranged, very similar to the Zika study, there was a placebo group, 2 doses tested, 5 times 10 to the 10th viral particles and 10 to the 11th viral particles. Turns out those 2 doses were basically the same. And we also tested a single shot vaccine in the lighter color and a 2-shot vaccine in the darker color. You can see that placebos, as well as the baselines, have a few individuals who are positive for binding antibodies. We think these are real. These are probably people who were infected in the spring surge. But the vast majority of people were negative. But by 29 days after the initial vaccination, the vast majority of individuals showed binding antibodies, and by day 57, I think it was 100% of individuals showed binding antibodies against SARS-CoV-2 spike. These then persisted even in the single shot vaccine group to 10 weeks at day 71. And in the groups that did get boosted at day 57, there was an increase of about 3 to 4-fold by day 71. So the summaries are shown here. Single shot vaccines are these 2 lower curves. And you can see that essentially everyone developed antibody responses. The groups that got boosted showed an increase of the antibody responses. And even the single shot vaccine shows very good durability for 10 weeks with really no evidence of decline. So although we can't really claim durability until we see the 6 and 12-month data, then this is certainly on a trajectory to showing very good durability, because there's essentially no evidence of decline at all at 10 weeks. Cohort 3 is very similar to cohort 1, except that these are elderly individuals over 65 years old. And you can see that virtually all elderly individuals also generated binding antibodies to similar titers as younger adults. These are neutralizing antibodies. To a first approximation, it looks like a very similar graph. Very few individuals in the placebo group or at baseline had neutralizing titers, only a couple. Probably reflective of past infection, but the vast majority at day 29, and essentially everyone by day 57 developed neutralizing antibody titers to this virus after a single shot immunization. In the single dose groups, this persisted with no evidence of decline through day 71. And in the individuals that were boosted, there was a boost of 3 to 4-fold in titer by day 71. So a very similar response is seen for neutralizing antibodies. Essentially, almost everyone develops neutralizing antibodies 4 weeks after a single shot vaccine. These titers are persistent and durable for at least 10 weeks and almost certainly much longer. With a single shot vaccine, and with a 2-shot vaccine, they can be boosted. And also similar neutralizing titers in the vast majority of individuals in elderly individuals in cohort 3. So there were also T-cell responses seen that I didn't have time to show you, as well as other antibody functions. The phase 3 studies started in September, and the first phase 3 study is a randomized double-blind placebo-controlled trial for the efficacy of this vaccine. This study was fully enrolled in December with an enrollment of about 45,000, and the results are, the database is actually being analyzed right now, and the results are going to be released. I was hoping it would be this week. It might be early next week, but within days, we're going to see the results of this trial. This was a 1-to-1 vaccine placebo randomization that tested a single shot vaccine at the dose of 5 times 10 to the 10th viral particles, with the features of it being a single shot vaccine and no requirement for a frozen cold chain in the United States and South Africa, as well as in many countries in Latin America, including Brazil. Now, when we see the results of this trial, then the first question is, does it show efficacy in the United States at all? If so, then we think it'll be relevant for a rollout. But also, is there going to be decreased efficacy in South Africa and Brazil? Because we know now that there are variants of SARS-CoV-2 emerging in South Africa and Brazil that at least partially evade neutralizing antibody responses. The second phase 3 study is still enrolling. It started in November. Target enrollment is 30,000. And it's basically the same study, except there's a 2-shot regimen at weeks 0 and 8, as opposed to just a single shot regimen. So a question that's really been dominating the headlines and the scientific interest over the last month or so is, will the new SARS-CoV-2 variants that I presume you've heard about, will they reduce vaccine efficacy? And some early data from several groups show that the UK variant does not appear to evade neutralizing antibodies, but the South Africa variant, largely because of the 484 mutation, appears to substantially or at least partially escape neutralizing antibodies, either in convalescent individuals or in vaccinated individuals. And whether this decrease of 6 to 8-fold of the neutralizing titer, whether this causes a clinical problem for vaccines or not, remains to be determined. So I'm going to close now with a couple perspectives, and then we have time for some questions. So vaccine development for COVID-19 is proceeding faster than for any pathogen in history. Safety and public trust are absolutely critical for any vaccine. We feel that multiple vaccines need to be implemented in parallel, since we need to accelerate the vaccine rollout both in the United States and in the world. Viral variants require further study and may reduce the efficacy of vaccines, and additional variants will likely emerge. So we hope that we're not going to be in a cat and mouse game with revised vaccines chasing a constantly changing virus. What we really need is to vaccinate the world as quickly as possible to reduce overall levels of the virus so that we actually prevent the emergence of further variants that could potentially even be bigger problems than the current ones. And finally, we have to realize that there's 7 billion people in the world. And so we therefore need multiple vaccines to be successful and simultaneously implemented throughout the world to end the pandemic. So these are US numbers, but the global numbers are even substantially worse. But with substantial numbers of cases and deaths every day, the short-term future is going to be extremely challenging in the next few months, and there's no way around it. But with the first effective vaccines now being deployed and hopefully more vaccines coming, the midterm future for this calendar year, I feel is hopeful. And the scientific accomplishment of multiple groups throughout the world developing COVID-19 vaccines in a year is a truly stunning scientific accomplishment that really exceeded anyone's expectations. And it really shows the power of science to help solve a global problem. And so with that in mind, I would say the long-term future remains bright. And so I'm often asked when the pandemic will end. And of course, I can't tell you that. But I do think that vaccines are going to be a very important part of it, together with public health measures, as well as all the other interventions that I'm sure you've been hearing of. And I think when historians look back on the pandemic, the global pandemic of 2020 and 2021, then science will be seen as critical for ending the pandemic. And so for you folks interested in translational medicine, I think that there could never be a more important time than today to see the power of science to transform medicine, to tackle a global health problem. And in the end, it's going to be a science solution to the pandemic. I'll stop there and happy to take any questions.
Thank you so much, Dr. Barouch. Wow, I'm stunned. Thank you so much for being so transparent and informative about your journey with COVID-19. It's incredible that it's just, as you said, just around the corner, just a couple of days out before, things can really change, or we could see a huge change. So that's incredibly exciting. So we might go to some of the questions there now, if that's all right, because there's a few coming in, or there's a few already been sent in.
And I'm happy to answer any questions to the best of my ability about any of the scientific issues. Since you guys are students, I can tell you more about anything you'd be interested in terms of a career in science, or what science means, or anything that you would like, really.
Brilliant, thank you so much. So I'll just go first with some of the questions already sent in here now. So one of the questions was, as you mentioned at the end, that there's that need to, because we need to vaccinate as many people as possible and as soon as possible. So we need to have many vaccines on the market. But how does that actually work in terms of these vaccines have different approaches, different mechanisms, but how are they treating the same thing then? And are they treating them to the same efficacy, I suppose?
Well, we know that the RNA vaccine, each vaccine is going to have pros and cons. And so there's probably no single vaccine that's going to be best by all measures. For example, we know that the RNA vaccines from Pfizer and Moderna, they are very effective. At least we know for short-term efficacy. We don't really know anything beyond short-term efficacy. And actually, we actually don't know whether they actually prevent infection. All we know is that they can prevent symptomatic disease. But we also know they have limitations. They're more expensive. So for a global rollout, they could be a problem. We also know that they require sub-zero freezing. The Pfizer one, even ultra cold freezing. And they also require 2 doses. So that might limit their use in some areas of the world. The efficacy of the inactivated virus from China looks like it's substantially lower, only about 50% efficacy. And so why that is and what are the immune correlates of that, I think we can learn a lot from science, but clearly that vaccine is not as effective. The AstraZeneca vaccine, quite honestly, the efficacy is sort of uncertain because of some complications in the trial. And so somewhere between 60 and 90% effective. And it's not completely clear whether it's closer to one end or the other end. I think additional trials are in progress. So there will be differences. And of course, for our vaccine, we have no idea because we haven't seen the results yet. So there will be differences in efficacy. In some cases, there will be differences in convenience. In some cases, there'll be differences in cost. For the Ad26 vaccine, it's being tested as a single shot and a 2-shot vaccine. And so a question even within our own program is, what difference in efficacy would warrant a single shot approach? because we know from mass vaccination campaigns that the simpler it is to vaccinate, the broader population you'll reach and faster. So the logistic advantages of a single shot vaccine that doesn't require freezing is more than just a convenience. It actually could be reaching a substantially larger number of people in a substantially faster period of time. Of course, if there's a major difference in efficacy, if the one-shot vaccine gives 20% efficacy and the two-shot vaccine gives 90% efficacy, just for an extreme case, obviously you go with a two-shot vaccine. If it's 75 and 85, then it's a much more difficult question, because with a two-shot vaccine, it's known clinically, a lot of people don't actually go back for their second shot. If you look at clinically how many people actually complete a vaccine series for hepatitis or other clinical vaccines, it's lower than 100%.
Okay, brilliant, thank you. And with that, and with many vaccines being available or hopefully available on the market, is there a way that you might know which one vaccine would be most suitable for you to take? Or is that something to take into consideration?
Well, currently we're in an era of vaccine scarcity. So there's simply not enough vaccine to vaccinate everyone at the present time. And so for the next period of time, it's not likely that people are going to be able to choose. Each hospital or clinic or pharmacy or whatever vaccine distribution center will probably only be offering one vaccine, because offering 2 to 5 vaccines would be a logistic nightmare. And so in most cases, people won't be able to choose. And that's simply because there's just not enough vaccines to go around, full stop. And so we're not really in a position where people can sort of order. Some people might want the Moderna vaccine, some people might want the AstraZeneca vaccine, but people really are not going to be in a position to choose because there's just not enough vaccine to go around to allow that. There might be some reasons, some medical reasons why one vaccine would be better than another. For example, if someone knows that they have an allergic reaction to a component of a vaccine like polyethylene glycol, that's actually a pretty hard reason not to take the RNA vaccines. Now that occurs only in a few people out of a million. But for people who have a known allergy to a component of one vaccine, that's a pretty good reason to get a different vaccine. And then ultimately, some vaccines might be more rapidly achieve an indication for children, for example. So there will be some reasons why people really would get one vaccine versus another. But I would say in an era of vaccine scarcity, where the objective is to vaccinate the world, then most people really should get whichever vaccine is offered to them most quickly. Okay,
thank you. We have another question here from actually one of our speakers that was speaking earlier on, who says that they had COVID recently. And he's asking, do you think people who have already been infected or young people in good metabolic health should still have?
Absolutely, because we know that the antibody responses following the vaccine are higher than from natural infection in most cases. And also we know that with natural infection, those titers do wane and people, at least to some extent, can get reinfected. So I would say if you were naturally infected, you probably have some degree of immunity, but it's not as robust as vaccination. And so the recommendation is for essentially everybody to be vaccinated.
And do you think that it is possible to have every individual, in the world to be vaccinated? And if not, are we still going to be living with the risk of the coronavirus, flaring up again with new mutations or anything like that?
I think that the best defense against persistent pandemic and the risk of future variants is to get as many people vaccinated as fast as possible. Yeah. Will we ever vaccinate every single person? Of course not. But I think that the modelers tell me that if we can vaccinate about 3/4 of the population, then that should do a very good job of achieving herd immunity. Okay. And of course, more than that would be even better.
Yeah, brilliant. Okay, great. We have some more questions here, but I think we have another session that's going to be taking place now at 2 o'clock. So Dr. Barouch, I just want to say thank you so much for joining us here today. Your work is absolutely incredible, and incredible doesn't really seem to cut it though, but it'll be really interesting to see obviously the future and in the next couple of days, but also it'll be really interesting to see the impact you have on, you know, individuals like myself or attendees here now who hope to, you know, pursue a career in advancing human health, and look to you for inspiration. So thank you so much, and we wish you all the best. Our fingers are crossed for the next couple of days. So thank you so much.
Well, thank you all for your attention, and I would encourage you to pursue your interests in translational medicine and to help solve global problems.
Thank you so much. Okay, it's wonderful to speak with you, and talk to you soon. Thank you very much.
Thank you.